All right, guys, this little mini lecture is going to cover carbohydrate digestion and absorption. Like all macros, digestion begins in the mouth with mastication, which is chewing, and then also the production of saliva. So, two different forms of digestion here. Remember, the saliva is produced here underneath the tongue and in the very back of the mouth. And with that, we have some salivary amylase. This amylase is the enzyme that's going to begin to break down our long chains of polysaccharides here into shorter and shorter ones. So we'll go from hundreds and thousands, and we'll continue to chomp them down all the way until we get into the large intestine. But back to the mouth here. When we swallow, we have the back of the mouth. This is the pharynx. Remember, we also have the epiglottis here, there to protect the trachea. But that food is going to travel down the esophagus through that involuntary but necessary peristalsis, that rhythmic-like motion, until it gets down to the lower esophageal sphincter. It's going to relax and allow food to pass into it. With this, we don't have chemical digestion that's occurring here because the acid that's produced here in the stomach, which you should know now, is hydrochloric acid. That should say HCl there. And that is going to deactivate the salivary amylase that was produced here in the salivary glands. So that's deactivated, but we'll still pick that up when we go into the small intestine. In the small intestine, of course, that's where the majority of digestion and absorption occurs. And with this, we have the pancreas. It's really hard to see in here, but it's kind of hidden by the stomach. It'd be right here. The pancreas is going to secrete bicarbonate that helps to neutralize that acidic environment. It's also going to secrete hormones like insulin and glucagon. And then last but not least, it secretes our enzymes that are going to help to really break down those long polysaccharides into dis, oligos, and also monos. When we get into the small intestine, though, we have additional enzymes. It doesn't show it here, I believe, but we have additional enzymes here that are secreted along the mucosal cells. So same types of things as we've been calling. We have salivary amylase, we also have pancreatic amylase, but we also have the brush border enzymes and amylase here that it's hard to actually draw those enzymes. Forgive me, I don't have the right equipment that makes this a lot easier. Okay, but eventually we're getting things down into smallest units here. You see the disaccharides, a mono, and you also see the oligosaccharides. When it gets into the smallest unit as possible, it will be absorbed into these mucosal cells. Once it gets into the mucosal cells, it can travel into the circulatory system, makes its way down and through, eventually meeting up to the liver. Now, anything that is not digested here in the small intestine will continue to travel through the small intestine into the ileocecal valve, which is here, not pictured, but it will go into here and it will make its way through this large intestine. Now, this is where we have bacteria enter. The bacteria is going to chomp down as best as it can. It can't get everything gone, but it's going to chomp down some of that oligosaccharides and fiber that wasn't digested, and it is going to make vitamin K, it's going to make biotin, but as a byproduct, it's going to produce gas, right? But the good part of that is it's going to also produce the short chain fatty acids, and those are very important in keeping the health and integrity of our gut. So a couple of things are happening here. We get as much as we can digested. Another thing that's going to happen, I want you to know, is that our large intestine is not going to do a lot of digesting and absorbing besides what we talked about as the indigestible oligos and fiber. But what it really does great with is that it reabsorbs some of that water that was lost and some of the electrolytes too. Without that, we would become dehydrated just from the mere act of digestion and absorption. So it reabsorbs some of that, and everything left over, I guess, eventually makes its way to the rectum and is excreted. All right, so we are in the absorption phase. We have been digesting it this whole time. We've gotten it down to its smallest molecule size. Now we have 3 of our monosaccharides listed here. This is supposed to represent the intestinal lumen space. minus this text. And then here we have the enterocyte or the mucosal cell. Here's the villi, which collectively with the micro is called the microvilli. Here's our three monosaccharides. We have glucose, galactose, and fructose. See that galactose and glucose, they both use active transport. It's active transport because it requires active energy. So ATP plus this carrier membrane, like a door. So both of these are the same. Fructose itself um, can diffuse nutrients in through facilitated diffusion. So we have a protein carrier here, but we don't have to have ATP for it. Regardless of the mechanism, it eventually gets into the enterocyte, packaged up, and it enters into portal circulation. Right, so this is all about the absorption part here. Now we have mobilization, right? We're going to transfer this, and our body is going to use it as needed. Eventually, all of these make it into the liver. Now, if your body does not have an immediate need for glucose, then it's going to try to store as much of it as it can. And by doing so, it's going to use condensation. Condensation, remember, it uses water in order to bond them together or bind them together. And now we have glycogen. Um, I'm sorry, with condensation, water is produced when they bind together from one mono to the next. All right, so now we have glycogen here, and this form or human storage form of glucose is just sitting there and it's waiting to be used when necessary. And this is necessarily used when you are not eating. So if you go longer than about 2 hours, your body's going to start pulling some of these glucose molecules individually off and sending them into really kind of back into your bloodstream in order to carry it to your brain. It's going to carry it to your heart, your lungs, whatever type of cell needs glucose in order to produce ATP or energy. So think about your heart. If your heart did not, as it being a muscle, if it did not have ATP, it could not pump. And so your body's constantly went all those ways to break things down into single units. We go to your liver, we store it back into a larger molecule like glycogen here, that's long branch chain, not amino acids, but branch chain polysaccharides until it needs. And when it needs it, it's going to break them all back down and put them right back into circulation. So it's constantly broken down, put together, broken down, put together until necessary. All right, so we have here this word glucose, that's your monosaccharide, but you don't see in here galactose nor fructose. And that's because majority of these two guys here are going to be converted into glucose eventually when it reaches here the liver, because it has to be in glucose form in order to form glycogen. So forming glycogen from glucose, stay with me here. We have this word, glycogenesis. Glyco relates to glycogen. Genesis relates to creating. So creating glycogen. Remember, if you dissect these words, it's going to be so much easier to get those test questions correct. Creating glycogen, so storing glucose as glycogen. Remember, your muscle cells also are able to store a limited amount of glycogen so that they can have an immediate fuel source of ATP when necessary. But also know that your muscle cells are stingy and they do not share. So if your glycogen stores were to become depleted in your liver, which can happen very easily, your muscle cells aren't going to come to the rescue at all. Let's see, anything I'm missing here? No, but I do want you to go ahead and just note this word, glycogenesis, because we are going to cover some other terms that sound very, very similar. But when you see these things or hear these things, break down these words because this will help you not to get them confused. Just slow down and break them apart. In prehistoric times and even in times of scarcity like the Great Depression, food was not always as available as it is today. We can go around the corner, we're going to find a gas station or fast food place and get our fuel. So our body adapted and it was able to find a way to take the leftovers that we're not immediately using and store it for later. So that's what our adipocytes are. They're fat cells. A technical way to say our fat. And so anything that's not left over, whether it be carbs, it's fat itself, or protein, if it's not used, it's stored for later. Even though today food is very abundant, our body still works in the same way. So let's say that we eat too many carbs. First, anything that's left over is going to be used in order to make glycogen. After we have fueled our liver and our muscle cell forms of our storage forms of glycogen, then once those banks are full, then we're going to go and convert it into a storage form. And that's what you're seeing here. This is a triglyceride. So it can convert anything into a fatty acid. Here's a glycerol backbone. That's part we have, what we have is carbons, hydrogens, and oxygen. If we just reform these into a different structure, these are all still carbons, hydrogens, and oxygen. We just combine extras, we make a triglyceride, and we store it for later. Um, our body's really, really good at storing extra energy. Unfortunately, it's not as good as actually getting rid of the extra energies. So we'll find out a lot more about this, but I want you to know that um, eventually our glycogen stores are full, and when it is, it's going to convert everything into this word triglycerides. We'll talk about this a lot in chapter 5.